Hello, everyone, and welcome back to Conversations about CVI with me, Professor John Ravenscroft from the University of Edinburgh. And today I will be having a conversation with Corinna Bauer from Harvard Medical School. Hi, Corinna. Welcome.
Thanks for having me.
It's great to have you here. So for those who have not watched any of these before, these are one-time takes. I have no editing software. I'm on my home Wi-Fi, so it's a little bit slow, so we might get the odd freeze and everything like that. So I do apologize for that. We're not professionals people. We're just people talking to each other using this media conferencing software. So please forgive us for all the ums and ers and all the reiterations that we might have to do. So, okay, so don't worry about that. So, Corinna, I just briefly introduced you there, but I wonder, could you please talk a little bit and introduce yourself?
Yeah, so hi everyone. As John mentioned, I'm Corinna Bauer. I am a neuroscientist or a brain scientist, and I work at the Massachusetts Eye and Ear, which is a hospital affiliated with Harvard Medical School in Boston. So what we do is we study the neural correlates of functional vision difficulties that are seen in some individuals with cerebral visual impairments, or the CVIs. So my colleagues and I use something called magnetic resonance imaging, or MRI, and using this we can look inside the brain and we can study the structure, the geometry of the brain, look at the pathways or like highways in the brain, and we can also look at brain function to tell us how well brain regions involved with vision communicate with one another. So by looking at all these different aspects of the brain, we can tell how the brain changes, and then we can correlate this with various aspects of functional vision in CVIs to get at the, the W questions, the hows, the whys, whens, wheres, that sort of thing.
Okay, I'm just going to take you back a little bit, Corinna. Just to help me out a bit, I think I understood some of the words. Neural correlates.
Yeah, exactly. Sorry, I thought about that.
What exactly are those neural correlates?
That was kind of like this key phrase in like neuroscience a little bit ago. So it's kind of like you are looking at the underlying parts of the brain that are responsible for a certain function. Yeah.
Okay, so it's those underlying parts then.
The underlying parts of the brain responsible for a specific behavior or something like that.
Okay, so as a brain scientist, okay, the brain is this big, huge thing. It's divided into all these sorts of like lobes and things like that, and cortices and subcortex. But which parts of the brain are you actually, as a brain researcher, as a brain scientist, which parts are you particularly interested in?
Yeah, so there are loads of different types of brain scientists, right? Some look at the individual cells in a microscope, but I use bigger techniques, and so we'll look at more brain regions as a whole. So as you know, vision takes part throughout the brain, and so what I'm interested in is looking at and characterizing the dorsal stream and the ventral stream, as well as some of those subcortical bits, so some of those parts that are in the middle. So specifically then we can go back and look at the specific highways. I don't know what the highways are called in the UK, if they're like interstates or something like that. But if we were in the States, we'd be looking at the big highways connecting regions, so the interstates of the brain, and also looking at some of those smaller county roads that go between the local towns, and to see how those change in kids with CVI.
Okay.
Vision. Yeah.
That's quite motorways, I think we'd call them.
Motorways.
Yeah. So, look, so I know you talk a little bit about the ventral and the dorsal streams, and we talked about that in other interviews with Professor Gordon Dutton. So I'm not going to go too much into detail, but I'm going to ask you how do you look inside a child's brain? Do you do like chop the head off, or what exactly? How do you do this?
No, we don't chop anyone's head off. I think the ethics would have a big problem with that one. No, so I don't know if anyone's had an MRI before or a CT scan, but it's just this big machine, and you lie on it, and you can watch videos or whatever. So you just have to lie there, and then using the physics of MRI, which I'm not going to get into because they're really really complicated, but you can look at the various aspects of the brain. So in like magic, through a computer program, we can reconstruct those motor ways that are in the brain. I don't know if that made sense, but
so it does. So so let me get you right. So you would put a child, any child. How old do you have to be as a child to go through MRI? Can it be any age?
clinically or in our research?
In your research.
Oh, in our research, right now we have ethics approval to go 14 years and older.
Okay.
But there are loads of studies who go younger. Sometimes you just have to train the child to be able to lie still for 20 minutes at a time or something like that.
Okay, and then the child can either watch a video or a program, and that then you can test various parts of the different functions of the brain. Then I suppose, is that right?
Yeah. Yeah.
So
Exactly.
So you can specifically target the ventral area, specifically target frontal lobe area if you wanted to, choice decision making, and you can target the dorsal area.
Yeah, totally. So if you are interested in targeting the ventral area, you could say, well, the ventral stream is responsible for object recognition, face processing, and those things. So we could have the child or the participant lay in the scanner. We would show them different pictures of faces or different pictures of objects. And then we can look at what parts of the brain light up in response to those categories. That would target the ventral stream. If it was the dorsal stream and we were interested in motion, we could show them moving dots, or we can show them moving figures and things like that, and look at how the brain responds to those different stimuli.
All right. Okay. So you can almost test for anything, and basically then once you've got the results, there's a analysis software, and it shows you which parts of the brain are firing or not.
Yeah. So it shows you what parts of the brain are firing or not, and it's always a comparison, right? So you would look at how the brain lights up when it's looking at faces compared to when it's looking at objects, or when it's looking to neutral faces, like with no emotion, or faces that are smiling or really sad, things like that. And then you can target the very specific regions depending on what comparison you're doing.
Oh, okay. So let's look at that example. That's quite interesting. So you might do an experiment, I guess, with a 15-year-old participant, looking at how they perceive emotion.
Yeah.
Sad, anger, disgust, surprise, all of those things. And so you would present a whole range of these emotions, either through the computer, I guess, and then you can see how, in fact, the brain is processing those emotions.
Exactly. Yeah.
All right. And then you'll be able to determine, against a typical control or a control within themselves, where there is, in fact, a deficit or a functional problem within that processing area.
Yeah, you got it.
Hey, I should be a brain scientist.
Yeah, exactly. Switch jobs. Just kidding.
So I'm interested then. Okay, so you've got the results. Okay, so our fictitious 15-year-old child, let's call them Gordon. Let's call the child Gordon, just for want of irony. So Gordon has a problem with emotions, probably anger, right? So difficulty in perceiving anger, or perceives anger through when people smile. He thinks other people are angry when they smile and things like that.
Yeah.
So once you get those results, what are you able then to, or I'm thinking about collaborative working. How are you able to disseminate those results to teachers, to pediatricians, to other doctors? Or do you just keep them for yourself and go, haha, that's really interesting?
So once we get enough little Gordons or, Amandas and Deborahs and Lindas, we can combine them as a group, and then we can, write some journal articles and some papers, or do conference presentations and disseminate the findings to the greater population.
All right.
But in terms of our research, because we like to make sure that we have a really good rapport and make a good bond between all of the little kids who are in our study and with their families. And oftentimes, parents are looking for validation for, why their kid might be seeing this way, or interpreting, angry faces or happy faces as angry faces. And so, of course, we share results with individual families just to make sure that they understand what's going on and what the findings potentially mean.
And do you find it difficult to get participants, or is it a reasonably okay in the sense that parents do come and go, please, could you do a scan on my child? Because MRIs are scary, right? You got these big things, they whiz around, they make a lot of noise, and you got your teeth filings falling out. No, you don't. I'm just joking.
No, you don't. No, you're gonna scare them. We get a mix. Sometimes you have parents and kids who, we can scan them again and again and again, and you can never learn enough. You can always learn more, right? And the kid's always willing to come back. But then sometimes you have parents who, their kids are already teenagers or young adults, and a lot of different processes, and so they might be hesitant to do a research study when it's not necessarily like a medical test, right? And then we also, on occasion, kids do get scared and they don't want to go in, and that's totally fine. There are other parts of our studies that they can do without going into the MRI scanner, because it can be scary. I went in myself, and I was nervous the first time, but I watched Call the Midwife, and that was great. But we have something called a mock scanner, which is just a pretend scanner. It looks the same, but it doesn't have any of the machinery inside. So you can go in and you can get acclimatized to it, and you can like kind of train yourself and build yourself up to it that way if you need to.
All right. Okay. Cool. So how much of the brain do you think you understand then? How much of, particularly with respect to CVI, how much do you think you've got a full notion of? There's a tricky question there. How much don't you know?
There's a whole heck of a lot that I don't know yet. It's one of those things where the more you find out that you have absolutely no clue about, because the brain is so complex. And, I look at it at the macroscopic, sort of top-down view, but there's so much stuff going on in those cells that I don't know anything about. And, and there's only so much literature that I can be familiar with. I think I know a little bit, but that's maybe a little bit more than other people, and other people know a lot more than I do, so.
Well, I tell you what, my knowledge of brain and CVI and all comes from your work. So, please.
Oh dear.
So, is there any? Let's take this opportunity. Is there any websites or resources that you might want to direct parents or researchers to look at?
In terms of CVI.
Yeah, and I can add them on as links and things like that. Apparently, I'm becoming a YouTube expert here, so so apparently I can add these links to various descriptions and things. So,
so one of the sites that I don't know if Gordon mentioned this or not, was CVI Scotland has a really great series on CVI and introducing individuals to the brain and how that relates to different parts of CVI. So that's one of the ones that I go to a lot, because I think, as a researcher, it's important to understand what the parents and the individuals themselves with CVI are experiencing, what some of their concerns might be, and those sorts of things. So I think that's one of my go-tos.
All right, good. Yeah, we certainly have tagged that up a few times now, so that's quite, particularly as I'm based in Scotland as well.
Yeah. I should mention, we also have you mentioned earlier about how do we tell people about our studies and our results. And we have a Facebook group for the lab. It's the CVI Neuroplasticity group or something like that. I'll send you the link and you can pop it up there. And you know, if we have different conference presentations or whatnot, we let people know through that.
Oh, yes, I was gonna ask you about that. I forgot about that. Again, another surprise question. For those who don't know, we obviously have communicated some questions beforehand, and none of the questions I'm asking are the ones that we have communicated. So I do say sorry about that. So brain neuroplasticity, what's that about then? I hear so much about it, you know. Is it able to summarize what it is within a few minutes?
Yeah, so neuroplasticity in general,
Yeah.
you know, so it's a term that gets thrown around a lot, and I think people tend to think of neuroplasticity as this like magic thing that's going to, for lack of a better word, and I don't like using this word, but like fix your brain, right? But it's really just a natural process, and it's like the brain's response to learning, or it's the brain's response to injury, you know, natural development. All these are different parts of plasticity, but it's essentially just the brain's changing throughout time. And you know, as we learn new skills, right? You know, when we were in elementary school and learning how to do our fractions, you know, and our brain was changing. That's an example of neuroplasticity. If we think about American football and all of the injury and the trauma that some of those players receive, their brain also changes in response to that. And that's also an example of plasticity. So it can be a positive thing, like when we're learning something and the brain is, you know, learning a new skill, or it can be a negative thing as well. And I think that's something that we don't really talk about much. It's not like a be-all, end-all magical formula that you know, will make the brain always better, you know.
Yeah, so it's something that you certainly can't rely on then. It's something that you can't, oh, don't worry, plasticity will take over.
Right. And I think, you know, there's definitely like plasticity, your brain can change a lot. Like it is a really wonderful thing. And you know, through training and through exercise and habilitation programs, you know, who knows, the brain may be able to take over and naturally gain back some of those functions that were potentially lost as a result of brain injury. But the extent to which that happens in CVI, we're not sure yet. And so I hesitate to I don't know, attribute things to neuroplasticity, just because then I think it's really easy to just rely so much on that. Does that make sense?
Yeah, it does make a lot of sense. I think what I'm getting at, and maybe I pushed you on this, is I too share exactly the same, in the sense that you can't rely on this, right? We don't know. We're doing a lot of work. Well, you're doing a lot of work on it, and other people are doing a lot of work. But at the moment, we don't know why. We don't know why in some cases plasticity takes over, and why in some cases it may look exactly the same kind of deficit, but we just don't know. So I think,
yeah, that's true. And you know, so I've been reading a lot of literature about neuroplasticity in general, and you know, there's not a ton of research done in humans who have had brain damage and looking at, you know, the extent of plasticity, but there has been a decent amount in animal studies. And, you know, some of them talk about that there's a limit to plasticity depending on the timing of the injury, the age at which that happened, the extent of it, and all this sort of stuff. And yet there are other instances where the animal or the individual recovers to near normal levels of performance. So, you know, it's really a balance, and where someone lies on that scale between, you know, plasticity, like beneficial, and a vulnerability, I think is a factor of, there are a lot of different factors that play into that, right? So there's your age, where the injury happened, when the injury happened, the amount of habilitation or rehabilitation that you do, and whether or not it's targeted to, you know, the specific behaviors. Even things like socioeconomic status, gender, all of those sorts of things all play a role. And I think we're really just starting to barely scratch the surface about all of that stuff. So
Gosh, fascinating, fascinating. So I guess one last question for any budding young researcher academic who might stumble across this is, how do you become a brain scientist? How do you do that?
um, yeah, good question. I don't know. You got to go to university. So I mean, personally, I don't know if I took a roundabout way. Like, I studied music and psychology in undergrad, and then I remember learning in the class about MRI, and I just being fascinated with it. And so, you know, went to my master's and my PhD, and all of a sudden I'm a neuroscientist. So I don't know.
But go to university, study
Study hard.
study
Listen
hard
to your parents.
and study I don't know, biology, neuroanatomy, I guess. Yeah.
Yeah.
Good. Well, is there anything you want to say or anything you want to ask me? I think that's probably only fair since I've drilled you on this.
All right. Well, how did you become an education person?
Well, that's fair enough. There's two answers. One is very simple. One is I got a degree in psychology. So my first degree is in psychology. My master's degree is in psychology and language. And then my PhD was in psychology as well. So I was working at the psychology department, and a job came up doing some research looking at children and vision, but with some psychological implications. And I said I could do that, and so I got a job then as an academic researcher studying education and vision, and from there I became much more of an educationalist, I think, and vision person. So yeah, I got a job.
So go to school, get a job.
Go to school, get a job, study hard, get a PhD. That's
Yeah.
right. Yeah. All right. Good. So it's a fascinating world. Is it a world where you go in and every day you're at the scanner, or is it a world where every day you have to do a lot of work, a lot of reading, a lot of understanding, not setting up the equipment, but understanding the kind of tests that you're going to be doing? What would be a typical day or a typical week? Let's put it a typical week.
Yeah, sure. So, you know, I think that all depends on where you're at in the study process, right? So like right now, you know, being stuck at home, I'm, you know, working on developing a new study actually with you. And, you know, so I'm at the phase where I'm just going through tons and tons of papers in the literature and trying to really understand some of the nitty-gritty bits that are going into, you know, the brain and the different behaviors that we're looking at. So then, you know, so that's one aspect of it. Another aspect is then, you know, writing grants and writing papers to let everyone know that what we've been up to. There's a part about, you know, recruiting participants, you know, reaching out to parents or having them talk to us and tell them a little bit about what we do. And of course, once we're at the scanner, we have to get the data and then analyze it, right? So that takes a long time. And that's really one of the main things that I'm responsible for, is actually going through and doing the computer, you know, making the computer go so that we know what's going on in the brain.
All right. Gosh.
Never said the same thing 2 days in a row.
Yeah. Brilliant. Well, excellent. And I'm going to end it there because I want to say thank you very much. Personally, I have to say thank you so much for all your hard work and your team. I know you're working with a larger team, and I just want to say thanks very much. I find your papers extremely informative and helpful, and so it's guided some of my thinking, and so I just want to say thanks very much.
Oh, thank you. This has been really fun chat and hopefully informative, and hopefully I didn't say anything too bonkers.
No, I'm sure you haven't. All informative. Great. Thank you very much.
All right. Thank you.